Okay. Good morning and welcome to our Chai and Why. So today's Chai and Why is a very special Chai and Why in the sense that our speaker is a very avid outreacher. She has been a very avid outreacher since she joined TIFR in 2019. So it's a pleasure to introduce Smitarupa Kahali. She joined TIFR in 2019 as a part of integrated PhD program of TIFR. And currently she is working with Professor Ankona Datta in the Department of Chemical Sciences. And they do a lot of interesting stuff. And she has done a lot of interesting work. And I think she's going to tell us some small part of it, which also perhaps is a part of a PhD thesis,
yes,
right? All right. So over to you, Smitarupa.
It's visible, right? The screen?
Yes. Yeah. Yes, screen is visible.
Okay. Hello? Yeah. It's not showing here. I'm in Zoom, but this is coming over here.
All right.
So, but is it visible, the upper part? Yeah, that's fine. Okay. So, yeah, hello everyone. So, today I'm going to speak about the self cleanup procedure inside our body. So, okay. One second, I have to, I think, stop share and share it again. Yeah. One second. Here, yeah. So our body is made up of different. I am in presentation mode. How? Hmm. I don't think it's fine. So, yeah, sorry for that. So, now it's fine, right?
Yeah, back.
Okay.
Come out of the presenter.
This one. Duplicate slides.
Yeah.
Check once.
Oh, go ahead.
Now it's okay?
Yeah.
Yeah. Okay. So, yeah. So our body is made up of different organs. And if we go deeper, then each organ is made up of tissues. And if we go deeper and deeper, then the fundamental unit which we reach are the cells. So, so cell is the fundamental and the smallest unit of life. And so our body is nothing but a group of cells working together. So different parts of our body has different types of cells, and they perform millions and millions of processes to make us functional. Like I am standing here talking about this, you're sitting and listening to this. So everything is possible because all the cells in my body are performing the way they should. Otherwise, it would not be possible. So now, inside the cell, uh, yeah, that is muted. Okay. So inside the cell, there are several compartments, which we call the organelles. So like the nucleus, mitochondria, Golgi apparatus. So in this compartment, so you can think of this as different departments which are responsible for different processes which are occurring inside the cell. So, so we can imagine the cell as a factory. Like the factory also have these different machines which are carrying out different processes. And similar to a factory, because in a factory you can imagine that if several processes are happening simultaneously, you will there will be waste generated inside the factory which needs to be cleaned up and recycled. There is a constant need of raw materials for these processes to happen. And also, if there are several machineries working, the machines will also get malfunctioned from time to time. So similarly, in the cell also, there will be waste generated. The cell will need a constant supply of raw materials, as in the nutrients, and also the cellular machineries, the organelles or different proteins or different machines inside the cell can get malfunctioned. So in a factory, you can see that we have now devised that we can there is a system of this cleaning up the waste and recycling, and there are particular systems in a factory. But similarly, inside the cell also, the cell itself has an inherent cleanup and recycle process which is going on all the time. So this process is called autophagy. So autophagy is derived from the Greek language, which actually means self-eating. So why self-eating? Because this process happens inside the cell, and basically the cell eats up its own waste and malfunction stuff so to degrade and recycle them. So it is called autophagy or self-eating. And it's a very important process which actually led to this Nobel Prize to gave Nobel Prize to Ishinori Oshimi in 2016 for the discovery of this very important fundamental process. So what actually happens during this process inside the cell? So whenever any protein or organelle or anything gets damaged or malfunctioned, or there is waste inside the body which needs to be cleaned up, then the cell, what the cell does, it encapsulates them inside a compartment. So it starts with a membrane and then it goes whole, and it encapsulates whatever it needs to be degraded. So it forms this we call this compartment which has encapsulated, we call it a vesicle. Now there are other types of vesicle, which is this red one you can see. So these are called lysosomes. So what is there in lysosomes? The lysosomes have this enzymes which are proteins. So what these enzymes do is that, in general, enzyme is anything which catalyzes some reactions inside a living cell. So lysosomes has this specific enzymes or proteins which actually catalyze the degradation of this waste material which has been encapsulated inside this vesicle. So what happens is that the lysosome merges or fuses with the initial vesicle which has formed, and ultimately it forms a larger lysosome, and this enzymes then degrades the malfunction proteins or waste material into smaller components. Like if it's a protein, then it will degrade it into amino acids, which are the fundamental unit of proteins. Or suppose if it's a sugar, then it will break down it into glucose, fructose. The smallest, it will break it down to smallest unit. And then the smallest units will be again released back into the cell, and the same things can be reused by the cell for making up, building up new proteins or new materials which the cell needs. So it not only helps in degradation of the waste, but it also helps in reusing them, recycling them for building newer stuff. So when does this process occur inside our body? So autophagy happens at a basal level at all times inside our body, because at all times some process is going on and some waste is generated, something gets damaged, and so it happens all the time. But when does this process gets more active, or I'll say it increases? So there are sometimes like if you are doing fasting or like this dieting and nowadays also people do this intermittent fasting and all. So what happens is that when you're fasting or it's a sort of a starvation, so your body is deprived of nutrients for some point of time. But the body cannot stop its functions. It has to continue doing all the processes which are going on. So what happens is that for doing those processes, the body, like I said, is just like a factory. So it will need the raw materials. And if your body is deprived of nutrients, it won't get the raw materials or the nutrients from outside. So what the body actually does at that time is that it can degrade the damaged proteins or anything which is, so it will increase the occurrence of autophagy so that it leads to degradation of some proteins or some macromolecules, which will result in the like giving us the smaller nutrients like amino acids or glucose and sort of thing. And those can be reused in these processes. So another time is that when we are sleeping, this process is highly activated because again, when you're sleeping, the body is not getting anything from outside. So if it's a longer sleep hour, then at that time, autophagy will be highly active inside the body. And also if you are exercising regularly, so that also makes this process highly active inside the body. So, and this is one of the reason why exercising or good night's sleep or having food at a particular time can help in maintaining our body. So now why I say that this process helps in recycling and degrading the waste, but how important is this process? Like how important it is to the process for this process to happen inside the body. So since it recycles and gives us the raw materials, it's obviously important to make this process happen. But also, since it prevents the waste material from accumulating inside the body, it will reduce the chances of many diseases, because diseases, like there are some diseases like neurological diseases like Alzheimer's, which can happen when these damaged proteins or like the proteins which are not should not be inside the cell, they accumulate inside the cell, that can lead to this type of diseases. So autophagy actually helps that because it degrades the damaged proteins. And so if autophagy is less, then it will lead to an accumulation of waste material inside the cell and can cause several diseases. Next, autophagy can slow down the aging procedure, because if autophagy is very well activated inside the body, then what will happen? The cells will degrade the damaged parts and make newer things. So it will be all the processes will be like in the way they are like in the beginning. But if the autophagy process is like down-regulated, it's happening in a much lesser thing, then what will happen that all the machineries, all the proteins inside the cell will get old. They won't be repaired or rebuilt, and that will make the aging process much faster. So an activated autophagy actually causes a slower aging. And also, it's very important in the immune cells. So immune cells are the defense, forefront defense mechanism inside the cell. So which actually helps us during viral or bacterial infections. So autophagy plays an important role in these cells because it also helps in the degradation of the pathogens like virus or bacteria using a similar procedure. So also an activated autophagy can boost our immune system. So autophagy helps in a lot of processes, like it boosts immunity, it slows down aging. It also plays a role in sometimes in tumor suppression. So it's a very important fundamental process. But like anything, like whatever is important for our body, it's important at some optimum level. Excess autophagy can also be bad. Like if it's overactivated inside the body, what it will happen is that it will start degrading the essential proteins or the essential materials inside our body. That will also cause a problem. So autophagy should happen not less, not more. It should happen at optimum level inside the body. So this process is very tightly regulated in our cell. And if any disruption happens in this regulation, it can lead to several disorders. Like if autophagy is happening very less inside the body, it can be related to this accumulation of different. Yeah. Where is my cursor? So it can lead to accumulation of damaged cells, premature aging, weakened immune system, as I said. And it can also lead to inflammations because it leads to accumulation of damaged stuff. It can also lead to impaired tissue repair. Like if there is any damage to any tissue, then also there's the repair of the tissue will be slow because again, for that we need this raw materials. And also, since there will be accumulation of this waste and damaged stuff, it will ultimately lead to the death of this cell. Optimum level autophagy will lead to healthy cells. But again, if autophagy is overregulated, it will lead to breakdown of essential components. It will lead to tissue degradation because it will degrade the essential stuff. And again, and it's related to cancer progression. I will come to that in a bit. And also activated autophagy can also lead to cell death because it will degrade out the essential stuff. So you can see both increase and decrease ultimately leads to death of cells. So now, as I am talking about how autophagy should be well regulated and both the things can be difficult, I want to share an interesting link between autophagy and cancer. So cancer cells have this property that they grow uncontrollably, right? So when they actually want to go and grow uncontrollably, so they will need a lot of nutrients because there should be a rapid cell division and cell growth. So for that they need a lot of nutrients. So cancer cells actually utilize this process of autophagy to get more nutrients so that they can grow faster or divide faster. So they use this manipulate this process to their own advantage, and they use enhanced autophagy for their own growth. So in that cases, you can think that maybe if I give a drug to reduce the level of autophagy, then maybe we can solve this problem. But again, if autophagy is so I will say that it's not very obvious that it can only lead to the killing of the cancer cells by decreasing the autophagy. Because if you also increase the autophagy very much, that also will will kill, can help in killing those cells. So both the things can also be used as like therapies, like both decreasing and increasing autophagy can also be used in a therapeutic way. But the problem is, as I'm saying, it's a very qualitatively, I'm saying that either increasing or decreasing. What is the optimum level? How much autophagy should happen in a cell for it to remain healthy, and when it will go back? We don't know. So if we don't know that, then it's a problem is how will we actually quantitatively treat or diagnose these diseases? So for this, we actually need to know more details of how this process is being regulated inside our cell under normal healthy conditions. So otherwise, because dysregulated autophagy has been related to almost all kinds of diseases you can think of, from neurological to metabolic diseases like diabetes, to cancer, to autoimmune diseases, and everything. Like you can look up the Google and search any disease and it's linked to autophagy, there will be a link to dysregulated autophagy. So, in order to, so our aim was to understand this process in a better way so that we can devise therapeutics or diagnostic tools to understand this process better. So during this process, as I've already mentioned, that it goes through three phases, where the first, there is the waste material gets encapsulated inside a vesicle. So that stage is called an autophagosome, because it's an autophagic vesicle. So from that, it's autophagosome. Then the lysosome comes and fuses with this autophagosome, and that stage is called an autolysosome. That is like merge between autophagosome and lysosome. And ultimately, it forms a bigger lysosome. So there are three stages you can see of autophagy. And these autophagic vesicles are very, very small. Like, so it's around 1 micrometer. So it's almost 10,000 times smaller compared to a normal fly or a mosquito. So it's a very small vesicle which develops inside the cell. And not only that, so this process, any process inside the cell is a complicated one, and it's regulated by a number of multitude of players which are inside the cell. And so similarly, this process is also regulated by several players like different proteins, different metal ions, molecules, protons, and everything. So if we want to understand how this process is regulated, we have to understand the biology of these regulators which are regulating the process. So because so if you can consider that it's like a play or a dance program, and so everybody has a role to play, everybody has different dialogue or steps to perform, and if anyone does a mistake, then the entire performance goes wrong. So similarly, if any of the regulators are misbehaving, then it will lead to a dysregulation of this process and cause disease. So what we need to do is that we wanted to look into this regulators of autophagy and see how they are behaving under both healthy and disease states. So how to do this? Because a cell itself is very small. It's around 10 to 20 micrometers, and we need a microscope for looking into the cells. And if you see just the image of cells from a microscope, it actually looks like this, where like it's a contrast image in grayscale. And so this is a complete cell, and inside there are like 10 times smaller vesicles, which inside that we have this different proteins or ions or molecules. So typically, in using a microscope, we can't look into the ions or molecules which are inside the vesicle or regulating them. So how can we actually get information about the regulators there? So it's a very crowded place, and so let me give an analogy. It's a crowded place where I'm walking somewhere in the crowd, you won't be able to look where I'm going, because it's not possible. The similar to the cells where I don't know where the regulators are going. But if I have somehow a red balloon in my hand, then by tracking the balloon, you can actually see where I'm going in the midst of the crowd. So our idea was that if we can use some sort of, if we give a like balloon sort of a thing to the regulators, then we can look into where it is going or what it is doing inside the cell. From that, we can get an information if it's behaving in a way it should, or it's behaving in a way it should not. So, similar to the balloon, we make these molecules inside our lab, which are called sensors. So, sensors, if you see the this purple one, is the reactive or the receptor unit. So, what it does is that this unit binds or reacts to whatever I want to detect. Suppose I want to detect sodium ions, so it will go and bind to sodium ion or react to that. And as soon as it binds to the ion or molecule of interest, the bulb will glow. So it's a fluorescent dye which we use. So the fluorescent dye only starts showing fluorescence or glows when the receptor or the reactive unit binds or reacts to the analyte of interest. So, and it is such designed, the sensors are such designed, we design it in such a way that is very selective. Suppose I want to detect hydrogen ions, and if sodium comes and binds or reacts to it, there won't be any change in this bulb. So we won't be able to see it. Only there will be a change in the fluorescence or the glow of this bulb when the analyte of interest is reacting or binding to it. So it should be very selective. So that we can actually spy on the analyte of interest we want to detect. So what we wanted to detect in this, in the context of this process, first analyte which we wanted to detect was pH, where it is nothing but the concentration of the H plus ions. So why pH? Because we have realized that the pH inside the inside the autophagic vesicles, so the autophagosome, autolysosome, lysosome, these are acidic compared to other parts of the cell. So if we can have a sensor which tracks acidic pH, or where the sensor, the bulb glows in acidic pH, then it will only glow in inside these vesicles, inside the cell. And wherever I will see that glow, I can infer that those are the vesicles. And depending on the number of bulbs glowing inside the cell, I can actually conclude about the extent of autophagy which is happening inside the cell. And I can track this under different conditions and see how different conditions are affecting the extent of autophagy which is happening inside the cell. So, so that's why we wanted to design a sensor which will track the H plus ions inside these autophagic vesicles. So we had a sensor with it has a red dye which actually increases fluorescence or glows when pH is decreased, that there is increased amount of H plus ions. And also, we wanted our sensor to go specifically inside the autophagic vesicles. Otherwise, it will happen, it can go to other places inside the cell. Or what can happen? Let me give an example. Like maybe 5 sensors are going to some part of the cell, and maybe 100 sensors have gone to other parts of the cell. Then also there will be some differences in this glow. The 100 sensors will accumulation of 100 sensors will give a more glow compared to 5 sensors. So that will also give 2 false results. So we also wanted to wanted our sensors to go specifically inside the autophagic vesicles, like the autophagosome, autolysosome, and lysosome. But how can we achieve that? So we can use a targeting group. So targeting group is a as a linker. So the entire thing is a molecule only. So a part of the molecule which will ensure that it goes inside these vesicles. So as a targeting group, we have used a peptide. So what are peptides? So amino acids, like a chain of amino acids give rise to a peptide, and this peptides folds to make ultimately proteins. So peptides are nothing but short parts of proteins, like a sequence of a particular sequence of amino acids joined together. So we used a peptide for targeting our sensor inside the autophagic vesicles. So now the question is, how a peptide can actually take my sensor inside the autophagic vesicle? Now what happens is, so if you see the vesicle, on the boundary of the vesicle, there are some proteins which are like the gatekeepers, okay? So which are called LC3. Now, what will happen? So when at normal conditions, autophagy happens inside of a body. So so any protein, any stuff can enter inside the autophagic vesicle. But the cell can't allow that to happen because otherwise it will lead to problem. So so it's like, so any protein or any material when it comes when it tries to enter inside the vesicle, these proteins or the gatekeepers stop them from entry because it does not recognize them. Just like if you're going inside of an institute, we have to show our ID cards to enter. If we don't have the ID card, they don't allow us to enter. So that's how. So if the protein has this tag or card, so this tag is nothing but a peptide sequence which gets attached to this proteins or whatever whatever the cell wants to degrade. So it gets tagged by this small peptides, and on seeing those tags, these gatekeeper proteins allow them to enter inside the autophagic vesicle. Now, what our idea was that we can use the same tag in our sensor, and that will allow us to enter inside the autophagic vesicle, because we are fooling the gatekeepers, because we may not have actual card. We are borrowing the card from someone else, and we are attaching them to us, and it's not seeing the face. It's just seeing the card. So that will allow the sensor to go inside the autophagic vesicle. So that's what we did, and then we applied our sensor to living cells and see how they are behaving. So I have, like for convenience, I have marked the boundary of cells. So this is a fluorescence microscopy image. So in the first image, where completely healthy cells, like normal autophagy is happening, you can see that there are some red dots inside the cell. So those are the autophagic vesicles under healthy conditions, which is happening. But when we starve the cells for 16 hours, you can see that the number of red dots inside the cell has increased significantly. So since our sensor is going inside the vesicles and only glowing at an acidic pH, we can conclude that whatever red dots are coming from inside the autophagic vesicles. And as the number of red dots are increasing, we can conclude that on starvation, the autophagy, the extent of autophagy, yeah, sure. Yeah. Yeah. No, these two are different cells. On starvation, the cells get stressed, so size reduces a bit. I agree, but it may not be like in all cells. But oh, these two are smaller, different cells. These two are not the same cells. So yeah. So I'm showing one cell. We have done this in several cells to conclude. Next, we use this rapamycin drug, which is used commercially for different cancer treatments, or it used for its anti-aging effects, and also it's used during organ transplantations to make the body accept the foreign organ. So this drug is reported that it can trigger autophagy inside the cell. So we use this drug. We treated ourselves with this drug to see if it is actually triggering autophagy or not. So similarly, when we see that when there is no rapamycin, it's completely healthy cells, then we can see very low, some few red dots inside the cell. But when we treat the cells with rapamycin, we can see an enhanced uh, red dots from inside the cell, and we can see an increased autophagy happening inside the cell. So similar studies can be done using this sensor under different other conditions, different disease conditions, and to see how the extent of autophagy is changing. Next, we wanted to detect hydrogen peroxide or H2O2 inside these autophagic vesicles. Now, the question is, why suddenly H2O2 and why not the other things? So H2O2 is a very reactive molecule, and it is an oxidizing agent, and it's present inside of a cell at low amounts all the time. And the amount of H2O2 actually fluctuates depending on what processes are happening inside the cell. So it has been reported, like there are several literature which say that at low concentrations of H2O2, H2O2 actually helps the cell in triggering autophagy and helps the cells adapt to stressed conditions like deprivation of nutrients or any other stresses. But if there is increased very high levels of H2O2, then it can it can stop the process of autophagy to happen and ultimately lead to cell death. And increased levels of H2O2 and dysregulated autophagy has been reported in several disorders. But we still do not know how hydrogen peroxide is actually affecting this process of autophagy. Is hydrogen peroxide present inside these vesicles? Is hydrogen peroxide levels changing from different stages of autophagy? Yeah. Enters inside the cell. Hydrogen peroxide. Okay. So yeah. So how the hydrogen peroxide is entering inside the cell, because we are not drinking hydrogen peroxide. So yeah, hydrogen. Yeah. Yeah. So hydrogen peroxide is actually produced inside our body. It's not like coming from outside. So one of the example is that so we have water. So water has H plus and OH minus. And in several processes, this OH minus can be oxidized to OH radical, and that leads to the formation of H2O2. So two OH radical forms together to H2O2. So several processes inside of a body. So several oxidation and reduction reactions are happening inside of a body all the time, and H2O2 is like a byproduct of those processes, or an intermediate of those processes. So so it's not for like comes from the outside, it's produced inside the body. No, what we are having nutrients linked to that H2O2 is not like there's not a direct link, I will say. But if everything, all the processes are working fine, it will be produced at certain steps of certain processes. But if they are not being produced, then that can also lead to diseases. And if it's suppose if it's produced in a very huge number, since it's a very reactive and oxidizing agent, that can cause also a problem. So I will not say that there is a direct link between what food we are intaking or to production of H2O2, but there will be an indirect link because the food we are having, the nutrients which we are getting from outside, those will take part in different processes which are happening inside the cell, and some of these processes will have H2O2 as the intermediate. So yeah, so there won't be a direct link, I'll say, but yeah, everything is linked eventually in an indirect way. So yeah, that's what. So there is again there was an indirect link that which people have observed that there is a link between H2O2 and autophagy, but we did not know how, because there was no evidence that if H2O2 is actually present in all the stages of these vesicles, inside the vesicles or not, or how it is is the level remaining the same in all the levels, or in some stage of autophagy, it's actually changing, increasing or decreasing. So that's why we wanted to detect H2O2 inside this vesicle. And again, we developed a sensor in the lab with the same targeting group so that it also enters inside the autophagic vesicle, but with a different dye, a dye which reacts to H2O2 and increases the fluorescence. So similar to the previous study, we have seen that when there was no starvation inside the cell, we do not observe much H2O2 inside the cell. It's very low. But when we starved the cells for 16 hours, we could see that there was an increase in H2O2 inside the autophagic vesicles, which were developed. And similar, we also treated the same experiment with rapamycin. And here also we can see at the basal level, when the cells are healthy, we do not observe much much levels of H2O2. But once it is treated with rapamycin, and we can so we can see an increased autophagy, and also we can see an increased levels of H2O2. So now our question was that, okay, H2O2 is there inside the autophagic vesicle. But the question is, we are not seeing it in at a healthy cell much. And also, there is a thing that we do not know at which stage of actually the autophagosome, autolysosome, or lysosome, which stage this H2O2 is is performing a an important role. So we had another sensor. So because using the previous sensor, the pH sensor, if you remember, the red sensor, so it only gives an increase in fluorescence when there is a low pH. So just by increase, this is a very qualitative thing, like increase or decrease. We can't actually identify the stages of autophagy using that sensor. So we had another sensor in our lab, which is also a pH sensor, but it has two dyes instead of one red and one green. So what happens is that, so at different pH values, these two dyes behave differently. So for example, at around pH 6.5 or 7, what happens is that the green dye gives a high fluorescence, while the red dye does not fluoresce or does not glow. So what happens is that the overall fluorescence which we are getting, overall glow which we are getting from these two sensors will be in the green. When we move towards more acidic pH, then the red dye starts glowing, while the glow of the green dye decreases. So at this level, at around pH 5 to 5.5, we can see both like some sort of glow from both the green and the red dye, and the overall intensity which we get will be in between red and green, so it's some kind of a yellow glow. And when it goes to very low pH, then the green dye stops glowing and the red dye starts showing much more glow, and the resultant glow which we are getting inside the cell is in the red. So as you can see, as pH is increasing, we go from red to yellow to green. So how will this will help? So depending on the color of glow which we are getting inside the cell, if we see that, we can actually identify the different stages of autophagy. So if we get a green fluorescence or green glow, we can infer that those are the autophagosomes. While if we get a yellow glow, those are the autolysosomes. And if we get a red glow, those are the lysosomes. So we did the experiments in live cells. So we did no starvation, 6-hour starvation, and 8-hour starvation, where you can see that in case of where the cells are not starved also, we can see a lot of autophagosomes which are being developed, but not much. You can see that the later stages of autophagy like autolysosomes or lysosomes. But when we starve the cells for 6 hours, say, we can see all the 3 stages of autophagy, where you can see the yellow glow, the green ones, and the red ones, like the autolysosomes, lysosomes, and autophagosomes. But when we starve the cells for a longer time, we can see that most of the autophagosomes have gone to the later stages, like autolysosomes or lysosomes. Now we employed the H2O2 sensor which we had developed in the same cell, so that we can compare the level of H2O2 in different stages of autophagy. So we did you can see these are the same cells which we have used in the previous sensor, and we can see some dots where we have higher blue fluorescence. And if we compare the two, like the pH sensor and the H2O2 sensor, you can see that the places we are getting a yellow glow with the pH sensor, we can see a higher intensity with the blue sensor. Okay, so and if you compare the other one also, you can see that the yellow places have the maximum intensity from the blue sensor. So from that, we concluded, and we had also quantified that the level of H2O2 inside the autophagosome, which is the first stage of autophagy, is very less. And but it increases to a maximum at the autolysosomal stage, when the lysosome actually fuse with this autophagosome. And then at the lysosome stage, it still remains significant, but it reduces from the autolysosomal stage. So, so this is the first time we actually could see that there is H2O2 inside the autophagic vesicles, and it's not remaining stable. It actually changes as autophagy progresses. Now, we still do not know why it is actually increasing at the autolysosomal stage, and it will be very interesting to further investigate why it is suddenly increasing at the autolysosomal stage. And since it is increasing, it should have some role in that stage. And if you can decrease that H2O2 levels in that stage, it can you can manipulate this process of autophagy. So how can this discovery help us? So first of all, we are understanding the process better. Like we can see that there is a link to how H2O2 may be linked to autophagy. And also, since we have another parameter, like we have this H2O2, so using this sensor, so under different conditions, as I say, that we can study in different disease conditions and see how the levels are actually changing. And if we know that how the levels are changing at different disease conditions, we can use these sensors for diagnosis of such diseases. And also, since we now know that there is a stage where H2O2 is surging or increasing, we can use that to our advantage to manipulate this process. So it helps in opening new therapeutic pathways to treat many diseases. So, so this is obviously more to be investigated in this regard, but it's a very significant step while understanding this regulators of this process of autophagy. So what we need to know, so we have now investigated this H plus and H2O2. We further want to investigate the other regulators of autophagy. So we get a comprehensive idea about how these regulators are actually regulating, how they are fluctuating during different stages of autophagy. So once we get a comprehensive view, then we will have a better, we will be equipped in a better way to understand this process, as well as to utilize this process for our benefit. So yeah, so that's what we have worked on. So we are investigating on this cell cleanup and recycling process, which is like happening inside the cells, and we obviously will want to investigate it further. So so if you want to know about this work in much more detail, so this is like a published article, and also it's been covered with press release by Eureka Alert. So you can go through that, or you can also contact us for getting more information, and you can also contact our group, because we not only we work on this process, we actually work on developing this sensor molecules for tracking different bioanalytes inside the cells for in context of several other important processes. So this is just one of the stories. So yeah, thank you. So yeah.
Okay, thank you.
Yeah, sure. Yeah. Can you be a bit louder? Yeah. Okay, yeah. Hmm. Hmm. Okay, so yeah, so the first question is related to how we suppose we take glucose or we take proteins from the outside sources. So how can we actually manage our lifestyle or diet to regulate the hydrogen peroxide inside our body? That's the question, like first question, yeah. So yeah, so that's a very interesting question, but I don't think we can actually have like specific diets for producing hydrogen peroxide or reducing the hydrogen peroxide inside the body. But yeah, if as I said, a balanced diet always helps, because balanced diet means that all the processes, like whatever breakdown and whatever rebuild is happening inside the body, it happens in the way it should be. So there is no direct link to the diet to hydrogen peroxide, but one question I can say that what I was saying, that we can modulate the hydrogen peroxide level to to manipulate this process of autophagy. So what we can do is that we can give stuff inside of a body. So hydrogen peroxide is an oxidizing agent. So, and it's also very reactive. So if we give we take something or a drug or anything, which will actually react with this hydrogen peroxide. So so if it reacts with hydrogen peroxide, so it will be changed to something else after reaction. So that will deplete the amount of hydrogen peroxide. For increasing the hydrogen peroxide, what we can do is that we can look into the processes which is actually producing hydrogen peroxide, the enzymes which are helping in the production of hydrogen peroxide, the oxidation or radical reactions. So we can improve those functions of enzymes. So we can indirectly manipulate the levels of H2O2, not directly by having some food or something. So so that's the first one. And can you repeat the second question? Okay. Okay. This acidic or alkaline water. Yeah, I got it. So you're saying that how acidic or alkaline taking water, will it help in the process of autophagy or the production of H2O2? So I'll say that the pH does not like when the process of autophagy is starting. So at that place, the pH is not playing a role. So once a vesicle is produced, like the first vesicle is produced, there are H plus ions inside that vesicle. And once you go to the different stages of autophagy, the pH decreases. So uh, if you take like acidic water, it can like decrease the pH of the entire cell. So that we don't want. We want like only the decrease of pH inside the vesicle. So uh, I I'm not sure, but I don't think having acidic or alkaline water will actually help in this process. What will help is that and another question you had, which is in linked to that, that will prolonged fasting will like cause problem. So yeah, definitely I'll say. So I'll say that to keep this process active, the best possible way is that have like good sleep, like 7 to 8 hours, which people say. And I think a lot of people do not follow that nowadays. Like regular exercising and physical activity is important, but I'll say we should not overdo it. Like even the fasting and the dieting, we should not overdo it. I think better to have a balanced diet or having food at regular intervals, so that like not like having food very like now I had something and 5 minutes later I'm having something. So there should be a proper gap between the intake of food, so that the body gets time to digest and degrade the waste products and everything. But again, if we have a very prolonged fasting or then it what will happen, it will like activate the autophagy process a much more higher degree, and that will create problem, because then the body will start to degrade the essential proteins or essential sugar or what is needed for the body to function. So that will create again a problem. So I think the best way to keep this process active is leading the life the way we should. But yeah, that's the problem. We are not doing that. So that's why maybe if there's any problem, then developing drugs and all will help. Yeah. Yeah.
Okay, let's do one thing. We'll get to online question. Take a break.
Okay, sure.
Just couple of online. What are the size of the cell physical we are looking at in this question?
The size of the cell we are looking at, it's around 10 to 20 micrometer. Yeah.
And secondly, how do the cancer cells expire?
How do the cancer cells respire? They respire in the same way like the normal cells do, but they do all the processes in a similar fashion like normal cells do, but they do it in a much faster way. Like so, I'll say respiration is a process which gives results in giving energy and everything. So I'll say the cancer cells do all the processes in a very similar way to normal cells, but it needs to do it a lot faster way. The rate is faster. So yeah, that's what.
So there is not bad.
No, no.
Okay, all right. So it's not unlike take a break. refreshment.
Yeah,
I
sure.
think she also needs a break. Okay, so thanks to Conference Call. All right. Guys, please help yourself with the